We're going to talk now about the synapse, and the synapse is the connection between two neurons. And there's two types of synapse. They can be electrical, which actually goes directly through what we call gap junctions between cells. But the one that we need to look at is the second one, which is a chemical synapse. And the chemical synapse is a communication between these two axons, and it is done via a neurotransmitter. And I've written the keywords for this story along the bottom here, so please make sure that you're familiar with what each of them means and the fact that you can use them. So we have an action potential coming along an axon, and as it gets to the end, it comes to what we call a presynaptic membrane, and this bulbous object shape is a presynaptic knob. And one of the ion concentrations which all cells keep is a high concentration of calcium ions outside the cell. And that means that there is a concentration gradient, electrochemical gradient, from outside to in. And at the end of the axon we have voltage-gated calcium channels. And what's going to happen is, when the action potential arrives, the change in membrane potential is going to open the voltage-gated calcium channels, and the calcium ions are going to come in. And what these calcium ions do is they cause vesicles containing the neurotransmitter, which is the chemical that's going to diffuse across this space, to fuse with the presynaptic membrane and eject their contents by a process of exocytosis. And so the neurotransmitter is exocytosed into what we call the synaptic cleft, and the cleft is the space. Now, we should say that a synapse called a cholinergic synapse, which is very common with muscles, has a classic neurotransmitter called acetylcholine. And this is the one that we would use as a standard example of a synapse. So the neurotransmitter in this case is acetylcholine. And it's going to be exocytosed into this synaptic cleft via the presynaptic membrane, because the calcium ions have been allowed to come in and have caused them to fuse with the membrane. Now, once the neurotransmitter is into the cleft, it starts to diffuse down a concentration gradient from a high concentration to low concentration to the postsynaptic membrane. Now, obviously a number of vesicles would fuse and you get a fair amount of neurotransmitter into this gap. One thing to say is I've blown this up hugely. The gap would be tiny, and the diffusion would happen very, very quickly indeed. And you know this is true because a nerve impulse and your response through nerves happens very, very quickly. So it's not a case of an action potential arriving, and then suddenly the neurotransmitter being diffused and it's slowly going across. It's such a small distance, a bit like oxygen diffusing into your alveoli from the lungs. It's very, very fast. And I'd like to blow up one of these acetylcholines and make it into a bigger circle here. Because what happens next is very important. In the action potential and the axons that we've learnt about voltage-gated channels. Calcium here, but we also know about sodium and potassium in the action potential itself. We don't have voltage-gated channels here on the postsynaptic membrane. We have ligand-gated channels. And ligands are a bit like substrates to an enzyme. They fit in to the protein channels here. And if one of these binds to the ligand-gated channel, it allows the channel to open up. And these are sodium channels. And so, as well as calcium ions being in high concentration outside cells, as we know through the sodium-potassium pump, sodium ions are in high concentration outside the cell, And the opening of these ligand-gated channels causes sodium ions to come in. So we now have positive sodium ions coming into through the presynaptic membrane and now affecting the potential difference of these cells, of the membranes. This is going to become more positive because positive ions have been allowed to move in. Now, we then go to the old question of an action potential. We know that around a place called the axon hillock on an axon, if a threshold potential is reached, then there will be an action potential as sodium voltage-gated channels open. And this is no different for this new neuron. If the amount of sodium influxing as a result of the ligand-gated channels opens, then if it reaches the threshold potential of about minus 55 millivolts, then the axon hillock will be the place at which an action potential starts. Sodium ions flood in from the rest of the outside because the voltage-gated channels are open, and then suddenly an action potential. And therefore the neurotransmitter has carried on the signal. That is a very simplistic way of looking at the axon and the synapse, because in truth it may well be that this one action of neurotransmitter opening these channels may not be enough to reach the threshold potential. It might simply contribute to the membrane potential. So if we think about our graph here, and we know that we get to about a threshold potential, then it shoots up, then comes down, and then you have a refractory period, whereas you've got hyperpolarization. We might just have, as a result of this neurotransmitter opening the channels, we might just have a slight increase. It might be enough to depolarize, and in that case, bang, we get an action potential. But the fact that it may not be enough is a hugely important feature of the synapse, because the synapse is able to offer a level of control to the nervous system in what is a very clear all or nothing response. An action potential, when it fires, will just go down, and it will always happen like this graph. And that means that the actual action potential itself offers no qualification of a signal. It's all or nothing, and that's a key phrase. And so what synapses are able to do is they are able to allow the nervous system to work in a certain way which really greatly enhances its, the way it behaves, to be more efficient in the way we want it to. So I'd like now to talk about the synapse, not in terms of just the physical process, but also about how it can do this job. So I just want to clear up one thing here, and that is that the ligand-gated channels will open when acetylcholine, if it's a neurotransmitter, binds. Now, in doing that, these channels are open. But we know from action potentials that we want a very, very clear stimulus to create an action potential, and then there not to be the contamination of repeat stimuluses when, or should I say repeat action potentials, when there are no more stimuluses. And can we see the danger here? If acetylcholine diffuses into the cleft and sets off these channels, then it may well be that the action potential happens. And there's still acetylcholine hanging around here, which might bind to other channels and create another action potential, when in fact we just wanted to respond to the first action potential coming. So we have a danger of repeat action potentials when actually no new stimulus has been created. And so we actually have a very clever system in the body. And it's this here called acetylcholine esterase. And this is an enzyme which comes along and breaks down the acetylcholine that's left in the synaptic cleft. And that means that just like the sodium-potassium pump and the leaky potassium channel reset the resting potential. This resets the system so that the only way that acetylcholine now can bind to the ligand-gated channels is if new neurotransmitter is released as a result of a new action potential. So it allows that specific impulse to be generated by a specific impulse and not the contamination of acetylcholine hanging around. The acetylcholine that is broken down is recycled and made into new acetylcholine, which is put into vesicles. So that's one way that we make sure